Hello and welcome back to Cell Division Mitosis with Mr. Schaub. Just a quick refresher from our last discussion. What are the four phases of cell division? Prophase, metaphase, anaphase, telophase. And what do we use to remember that? Pretty men always talk. Alright, here's a quick synopsis of prophase. It's the first stage. Our nuclear envelopes started breaking up, our chromosomes condensed, so on. Then we had, what was next? Metaphase. We remember metaphase because they line up in the middle. Middle. The chromosomes line up. Remember, they are not paired. Pairs do not line up, and that'll be important when we start going through meiosis. They all line up individually. So in humans, how many would we line up in the middle? 46. Excellent work. Okay. And now we are moving into our third stage of mitosis. What is the third stage? Anaphase. What is that? What's the acronym again? Pretty men always talk. Anaphase is the always part. Here we go. Let's check it out for a moment. Here is anaphase. What are the unique features that happen in anaphase? Let's see. Okay. In anaphase, we start having our spindles shorten. Remember, the centrosomes went to either side of the cell, have their spindle fibers connect to the center of the chromosomes. They are now splitting those chromosomes in half. Oh no! Do we only have half the genetic information then? No, because remember way back when we were going through DNA, from DNA to chromosomes, DNA started as a double helix. It double wrapped around the histones, which made the chain, the histone cord, which then wrapped around a protein and made one chromatid. And then there was a copy of that chromatid next to it. The two copies made a chromosome. So in essence, this one right here is the exact copy of this one. So are we losing some data? No. But is this one from mom and this one from dad? No, they're both from mom, and then their partner would be down here. This one would be from dad. You get a complete copy here and a complete copy here. Well, the blue ones are for mom or the green ones for mom, either way. You just understand that this one partners with this one normally. They're the same. They're like both chromosome number 5 or whatever. Okay, so first we have the spindle shorten. And what that does is it starts pulling that chromosome apart and pulling it to either end of the cell. Okay, so it's pulling to both poles. You keeping up with me, brother? Okay. You on the site? All right, just make sure you're following along. Go ahead and close that. You don't need a pinnacle right now. You don't need a pinnacle right now, dude. All right, so now these spindle fibers are shortening, pulling the chromosomes to either side. Also, the chromatids are then being pulled to opposite ends of the pole. Great. So we have two complete copies of the DNA, one on either side. Any questions? So far, so good? All right.
Is this the one?
Yeah, that's perfect. Thank you very much.
This is the big one?
Yep.
Okay, there's another website, Folded, about when this doesn't happen right.
Folded? Oh, yep. Oh, yeah. Thank you. All right. So after anaphase, we are moving into what's that fourth stage of mitosis again? Telophase. That's right. Like a telephone. All right. Now we enter our fourth stage of mitosis. Telophase. What happens in telophase? Let's take a look. It's getting crazy here. All of a sudden, the spindles are no longer there because they've done their job. They pulled sister chromatids to either side of the cell. Notice, we have one chromatid here, one chromatid here. One chromatid here, and obviously we would have, so how many chromatids would we have in either side of the cell? 46, right? Excellent. So both sides of the cell have 46 chromatids. New nuclear membranes start to form around the DNA. What's that going to become? The new what of the cell? The The nuclear membrane, which is going to make cover the nucleus. The chromosomes at this point now start to uncoil. Okay, they start leaving that protein scaffold that we talked about and becoming just a cord. Nucleosome cord. Do you remember that way back then? Good. You feel you with me, brother? All right. Okay. And then cytokinesis begins. Cytokinesis is the pinching off. Notice how the cell wall is starting to come in here and here. Why do you think that is? What's going to happen? That's right. Those two sides are going to come together and split in two. But we're not going to go into that yet because that's not part of my telophase. That is the last phase of our cell's life. All right. So any questions so far? Prophase, metaphase, anaphase, telophase. Remember, you're going to need to be able to say the names in order. You're going to need to know the names. And you're going to have to recognize which picture is each phase. Pretty men always talk. Prophase, metaphase, anaphase, telophase. Okay, so now let's go. Let's see where we are in our cell life. So we started out as a baby cell. We grew for a little while. We copied our DNA. We grew again. We got old. It was time for us to split. We went through prophase. Right, our nuclear membrane broke up. Our DNA condensed. Went through metaphase. Our chromosomes lined up in the middle. Went through anaphase. Our chromosomes are pulled to either side. Went to telophase. Our nuclear membrane is now forming. And ready, starting for cytokinesis. So now, let's go through the last phase of our cell's life, cytokinesis. Excuse me. All right. Okay. Let me get my notes straightened up here. So then, there we go. All right. Ready? Here it comes. Boom. What a buildup. Cytokinesis. The last stage of a cell's life is cytokinesis. The fireworks already happened. This is just kind of the cleanup. You guys ready to see the cleanup? I knew you would be. Here is cytokinesis. Cytokinesis, notice this looks very similar to what stage? Telophase. Because it is telophase. But from telophase, we go into cytokinesis. The end result of mitosis and cytokinesis are two genetically identical cells in place of the original cell. Two things are different. They have one chromatid for each chromosome. They don't have a full chromosome. They are smaller. The cell was twice as big. Now it's half as big. It goes cut in half, right? Makes sense. Okay, so this cell is identical to the what we call the father cell or the mother cell, the original cell. Okay. Now this is back to the baby stage. So now we are back to this spot. Right here. Well, let me get my there it is. We're back to this spot. Now both of those cells are going to go through, they're going to grow for a while, and then they're going to synthesize. What do you think happens in this synthesis stage? That's right. That's where we go from one chromatid to two chromatids. Okay, this is where it happens. I didn't explain that earlier because I didn't know if you know what I was talking about. Okay, and it's not until we go through mitosis that the two chromatids are split. Why is it important that we keep those chromatids the same? So every cell has the exact same DNA. Beautiful, beautiful. Okay, ladies and gentlemen. So now our two cells are on their journey through life, growing, synthesizing, growing, and going through mitosis to make two more cells, and on and on and on and on. Now remember, not every cell does this. Not all cells do this at the same rate. We talked about what cells do we say don't hardly do it at all? Nerve cells. That's right. What cells are doing it all the time? Skin cells. Beautiful. All right. Any questions before we stop? You guys are good? Here's the money. I'm going to stop it. All right. Goodbye, everybody.